from Dr. Guangping Gao. We've heard that he's been working in this space for a very long time. So really honored to announce him. And he's going to talk about the development of tissue-tropic AAVs. I'm looking forward to the talk.
Hi, everyone. My name is Guangping Gao from UMass Cheng Medical School. Thank you for the organizer gave me opportunity to share some of our work here. I must have my disclaimer. That is, I'm not. I know very little about CMT, and this is a great opportunity for me to learn, and very encouraged by all the progress made, not only in gene therapy, but also other modalities for treating this devastating disease. So here's my disclosure. So what I'm going to talk about is about tissue tropism of AAV gene therapy. So AAV, it's really a teamwork inside out. The capsid itself is very important, and it determines biology, tropism, as well as immunogenicity. And so for this reason, capsid is very centric for gene therapy development, particularly if you want to engineer tropism. So as you know, there are 4 different categories of methods for capsid engineering. So today I'm just going to briefly share two methods we tested in the lab. And so first one, everybody probably know that Jim Wilson and myself really start discovery of natural AAVs from primate tissues. So in this case, we just simply designed primers that amplify capsid sequence as a capsid is centric to vector engineering. And this is the first time use PCR-based methods back in 2000, early 2000, to amplify capsid sequence. By using this method to amplify variable regions of the capsid, we are able to discover over 130 of different capsids. And so particularly the one I think today I heard several groups mention about the AAV9, and this is probably most popular AAV for CNS gene therapy. And this was discovered based on my notebook, January 21st of 2003. And basically, one of the important, I'm showing you here, basically the PCR bands and the clones of the AAV9. And the most attractive to or relevant to this meeting is AAV9's capability to cross blood-brain barrier and transduce the CNS tissues. And so that's the natural capsid discovery. And of course, we continue doing capsid engineering, and this is one of the projects in the lab where we're trying to introduce rational design into the capsid engineering and also semi-evolution process. So in this case, we kind of cloned over 1,000 cell-penetrating peptides onto AAV2 backbone. And we're trying to see whether we can enhance AAV2's capability for ocular gene delivery, and after intravitreal delivery and migrate to photoreceptor cells. So in this case, we basically did 2 libraries. One is ubiquitous promoter to driven expression. Another is GRK promoter for photoreceptor specific evolution. And as you can see here, by using this strategy of a photoreceptor specific promoter, we are able to identify enhancement of a photoreceptor targeting after intravitreal delivery. And here is, once we have leading candidates, we express EGFP. And as you can see, we use AAV7M8, which is a benchmark so far, the best vector to cross to transduce photoreceptor. And then what we have done here is by do this screening process of a cell penetrating peptide. As you can see, we dramatically enhance transduction after intravitreal delivery, and as well as a photoreceptor. If we use a specific photoreceptor promoter express EGAP, you can see the enhancement. So this is really a proof of concept that combination of rational design as well as evolution, you'll be able to get more target specific capsids. So that is about the capsid. As I said, that's one of the strategies to engineer tissue specific tropism. But as many presenters presented today, and tissue specific, cell type specific expression is very important to accomplish tissue tropism. So in this case, as I said, AAV is a teamwork. So vector genome is very important. Determine the gene therapy efficacy and specificity, and avoid the off-target activities. So I'm just, in this case, I'm going to share a AAV gene replacement therapy for SMN or SMA disease. In this case, it's leading by my colleague, Professor Jun Xie, and 2 postdocs. What they are trying to do is, everybody knows Zolgensma is a very effective drug for treating SMA. But what are they trying to do is trying to make it better, or trying to make it better and safer, because as you know, Zolgensma causes some liver toxicity, and in some cases need to lethality in clinical patients. So the strategy they're trying to say is, if we could enhance the expression of SMN1 gene, and so we can reduce the dose, and by reducing dose, and then we make it safer and better. By doing so, they are really engineering the vector at a promoter levels, and also codon optimization, all different strategy. And by doing so, you can see they can enhance expression about 11-fold compared to benchmark vectors, which is Zolgensma expression cassette. And so from here, they choose the benchmark vector and their most efficient vector, and then generate AAV, and initially they inject the Zolgensma-like vector into the mouse, and found the animal only can survive about 11%, 20% for 90 days at different doses. And then they said, okay, after we optimize the vector, whether we can improve this. And what they found is, after they have this 11-fold stronger expression, they actually kill animal faster. And bottom line is here is basically both benchmark vector and their highly efficient vector will kill the animal. And so, of course, I don't have a chance to get through, but of the more details about it, everybody can think about it. This could be a liver toxicity. So to solve this issue, what they decide to do is go through not only tissue cell type specific, but a gene specific promoter, which is SMN1 promoter. And they found in vitro, this promoter expressed as strong as chicken beta actin promoter. And in vivo, with this vector, just simply change the promoter from chicken beta actin to this SMN promoter, and they were able to accomplish 100% rescue and beyond 90 days. And also there's a clear dose response, and also animal weight growth is significantly changed as compared to the benchmark vector. And of course, without saying, the motor function test in early stage and later stage both significantly improved. And so another important reason, as everybody talk about here at the meeting, is a biodistribution and the expression profiles of the transgene in different tissues. So our expectation is, if we use a gene-specific promoter, we should be accomplish Y type or at least heterozygous mouse expression level, as showing here, that's exactly what we saw. But if we use a chicken beta-actin promoter, then you have overexpression in some tissues that may cause the toxicity. And of course, by using this gene-specific promoter, we are able to eliminate liver toxicity. If we use chicken beta-actin, as you can see, we have a service liver pathology as well as ALT and AST elevation. But use this promoter, we are able to eliminate the liver toxicity. So in summary, and now with this, we call it second generation vector with gene-specific promoter, we are able to not only extend the lifespan and also reduce liver toxicity and the motor function and also other toxicity, peripheral tissue risk, everything is improved. And so that is the first demonstration about how important the gene-specific promoter for transgene efficacy as well as the safety. But just like Scott and Afruz presented, another strategy of gene therapy is gene silencing. And here, Professor Jun and his postdoc decide to test if we can use this neuronal motor neuron specific promoter SM1 to deliver gene silencing for SOD1. And as you know, this is a very devastating disease, and the animal model only can survive a very limited amount of time. So by doing so, as you can see, showing here, the survival curve is dramatically improved after using those different versions of a promoter of the construct driven by SM1 promoter. And also, importantly, we saw a remarkable delay of disease onset if we treat at neonate stage, and the delay can be 62 days up to. And then we start looking into, just like Scott showed, and what is a gene silencing, and not only in not only in motor neurons, you can see the yellow signal is SOD1 expression, but after using this vector, after gene silencing, you see dramatic reduction of the yellow SOD1 staining, and also the integrity of the motor neuron improved. And if you look at the body distribution, the silencing throughout the different body part of the body, you will show it's not only silenced in motor neurons, but also some other peripheral tissues. And of course, this will need to the functional approval by measuring plethysmography, measuring respiratory function in the animals. And also, we want to see how late could it be too late. What is the therapeutic window for this motor neuron specific gene silencing? As showing here, at the very late stage, when symptomatic stage started, we do the gene therapy, or even 1 week before it's dying, we're doing gene therapy. In both cases, we can expand the lifespan for the animal. And also, if you look at late stage treated animals, their growth curve is kind of somewhat normalized compared to without the treatment. So now I shared with you how to engineer an AAV tissue tropisms by either capsid or promoters, and for gene silencing and gene replacement. Now I'm going to end up with sharing you my personal and professional journey in gene replacement therapy of Canavan disease. And this, unlike this CMT1 disease, this is the more CNS disease. And however, the common point here is that both involve is myelination. Both are myelination disease. So this disease was initially discovered by Dr. Canavan back in 1930 in Mass General Hospital. And this is leukodystrophy. It's really myelination disease, and patient usually died before 5-year-old. Very devastating pediatric neurological disorder. And this is back in 2013, I went to a meeting in Orlando and met some Kahneman patients, about a dozen of them. But about 3 years later, I went back to the same meeting. Most of the children already probably passed away. So every time I see this picture, I remind me as a gene therapy researcher to develop gene therapy to save their life and improve their quality of life. And this disease, as we said, is a myelination disease. It's, as you can see, it's a spongy degeneration of the white matter in the brain. And this is a Swiss cheese structure because of a spongy generation caused by this an enzyme deficiency I'm going to mention about. Basically, my late mentor, Dr. Ruben Matanen, discovered initially a biochemical defect of this disease. What happened is that the NAA was made, this is one of the amino acid, was made in neuron, mitochondria, and transported to oligodendrocytes, get digested by aspartate. But when you're missing this aspartate enzyme, and then you have the NAA accumulate, you have multiple causes because of accumulation of the NAA, and particularly impact on myelin synthesis. And by doing so, you cause Kahneman disease. So I started this project 35 years ago, 1989, and I published the paper 1993, discovered the gene and the mutations of Kahneman. This made the gene therapy possible. So, and then afterwards, I joined UMass trying to develop a gene therapy. This is my MD PhD fellow, Dr. Dominic Jessler. And what he has done is basically optimize its expression cassette. As some of you talked about how important the expression cassette optimization. By doing so, he was able to enhance expression tenfold, fivefold, and reduce NAA accumulation, which is a biomarker for this disease. And then after we inject into the animal and through a first, we analyze, as you can see here, this is a myelin staining, and this is the wild type, and this is disease. And when we section the brains, you can see demyelination or whatever vacuole formation or hypermyelination and swelling axon. And 10 weeks, 4 weeks after gene therapy, this the first is 1 week after gene therapy, and 4 weeks after gene therapy, you can see that normalization of the myelin. So this tells you we can reverse the pathology and through remyelination and treat the disease. And also, if you measure NA in the brain by MRS, you can see a quick normalization and stabilization basically in 2 weeks, you drop to normal level. So this is very effective. And also, if you use the neural metabolic imaging, and you can see that basically, and this is a pathology you will see in Kahneman disease. And if you measure, use this multi-method to measure NA in the brain, and this is disease animal, and this is a disease treated with a control vector, and this is a wild type, this is a gene therapy animal. You see basically normalization of the disease. So, and then we first did a clinical trial, and not clinical trial, I mean, first in human collaboration with Dr. Barry Byrne, and this is a first in human trial, and you can see a reduction of NAA and normal growth and no LTST, and you can see improvement of the neural image. And finally, I'm going to end up here, tells you this gene therapy really started with discovery. First 62 years, we discovered the gene. And then next time I went to UMass and discovered AAV9 and start testing. And then we take 4 more years, basically put this in through the clinic. Totally, this is 86 years of the effort with the 4th generation of the scientists. And now, importantly, this is a clinical trial ongoing with Dr. Florin at Mass General Hospital, Florin Eckler at Mass General Hospital. So I'm going to end up with share a video patient share with me. This is before gene therapy, this patient. And this is 10 months after gene therapy. First time saying mom. And start walking with walkers. And this is 20 months after gene therapy. And start playing soccer. And first time stand up. And start walk first time. And walking through clinic. Swinging. And swimming, get off the pool. Say the first, I love you to the mom and dad. Start counting. So I'm going to stop here. Thank you for your time. I just hope one day we'll have same kind of therapeutic benefit to our CMT patients. I trust that's not going to be too far away. Thank you very much.